Hello, Zoology 2700 enthusiasts. This is the pre-lecture for Wednesday's lecture. Kind of sits in between Monday, where we talked about metazoan origins and porphyrians. We're going to keep talking about sponges on Wednesday and move on to placozoans. And I've got a couple more cool things that I wanted to talk with you about with the porphyrians that we don't need to be kind of face-to-face because it's largely just me talking to you. We'll save that time for doing active things like annotating graphs and that kind of thing. Now, the last time that I did this, it was much longer than 5 minutes. So, here we go. Sponges, remember, are captured in chapter 6 of Brusca. So, do that continuing to graze thing where you look up some of the same terms and you see some of the same diagrams that I'm using and hit on those high points of that text. One of the cool things, sometimes sponges, people get overwhelmed with the drill down kind of a narrative that we start out with in terms of the cellular types, and they kind of lose the big picture of seeing some of the incredible things that sponges can do. And sponges can do incredible things. And in fact, they affect human health. This is kind of a neat example where there have been byproducts that have been derived from Caribbean sponges, and this has been done for 4 to 5 decades. So Cryptotheca crypta, doubly cryptic in that naming in the species epithet of genus and species, was the source in the 1970s for cytarabine, which is a marine-derived anti-cancer agent that has been used. It's a common drug that's used for leukemia, chemotherapy, and involved in the treatment of HIV. So sponges, not just cool to look at, not just creating feeding currents and looking at asconoid, leuconoid, synconoid, those kinds of things, but actually being used and depended upon by humans today. Now, in terms of beauty, when we think about sponges, when I think about sponges, we've got to talk about these spicules. So the spicules, remember, are created by the sclerocytes. So in that diagram that you saw on Monday, where we looked at the cross-section, we talked about choanocytes and archocytes and porocytes and all these different cellular types that comprise that cell, that together make up the sponge. The sclerocytes are the ones that the founding cells that build spicules, and they can build them out of calcium carbonate, silicon dioxide, and they create beautiful forms. You're looking at different spicules here, scanning electron micrograph images of spicules that have had the mesohyal or the tissue digested away. And for years and years and years, taxonomists have used these species-specific morphologies of the spicules to help us differentiate which species is which. So they're critical to taxonomists. They're also critical to the sponge themselves. Now, if you've lived on a coastline, if you've visited a coastline, if you can imagine in a post-COVID future visiting a coastline again, you know that sponges, particularly sponges that exist in intertidal zones, require a lot of stiffness, sponginess, to rebound from the enormous forces of waves pounding them into the shoreline. And the sponge principally derives its stiffness or its sponginess from these spicules. And this is just there's a whole bunch of detail, but some extra links that you can go and jump into later on if you're actually interested in this. What is this? This are your kinds of engineering solutions derived from biological solutions. So these are looking at different cross sections of sponges and the different physical parameters of each one of these sponges. So you're looking at a cross section down here where they're individual components, so spicules contained within spongin that give the sponge its sponginess and allows it in a long, slender structure so that it can be particularly stiff and rigid. And there's a whole bunch of different ways that you can imagine using what we learn from the creation of spicules in terms of the design of tent poles, columns, stents, even in terms of using them inside of our body. So you're looking at a video here from this particular study here. So this is a Nature study that came a couple of years ago. And the last thing that I want to say about spicules involves the glass sponges, which we're going to talk about on Wednesday. These are some of the oldest animals in the world. You're looking at a single long spicule that this researcher is holding on the left-hand side. And these are laid down in rings. And you're looking at a cross section there in the center of this spicule, and the isotope, different isotopes of silicon within those rings can give researchers different clues about the silica concentrations that were present in the seawater when the sponges were growing, and that in turn can tell us about conditions in the ocean, but also when they were growing. And we know some of these glass sponges were alive 5 to 18,000 years ago. That's an individual organism that was alive 5,000 to 18,000 years ago. So sponges off the coast of British Columbia were alive when humans arrived in North America. So take a moment to consider the sponge. Some of them are older than we could possibly imagine. Some of them we derive cancer-fighting agents from, and some of them may provide biomimetic solutions to perform more efficient stent surgeries and even a better tent to take with you on the weekend. 5 minutes and 30 seconds. So we'll see you on Wednesday.